Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base Biotech podcast, and this is number 18 going out on July the 18th. It's International Nelson Mandela Day, and it's Friday. Another pretty busy week of news that we will get to shortly. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments other than please get off the air, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in sponsoring one or more podcasts or advertising with us. Last week we talked about a silica nanoparticle gene delivery system and a point-of-care diagnostic system for urinary tract infections. This week the subjects are neutrophil extracellular traps, otherwise known as NETs, with Citryll CEO Eduardo Bravo, and antimicrobial resistance with AMPLY Discovery COO Dermot Tierney. Alright, first let's go over some of the news headlines of the biotech news from the past week that you may have missed over at deeptechdigest.com. Baxdrostat met its endpoints in a trial in patients with uncontrolled or treatment-resistant hypertension. Emerging viral threats have been combated by a new dual lipid kinase inhibitor, and the Wecker Biotechnology Center opened in Munich. A new consortium is looking to deliver a fully automated robotics CGT manufacturing platform. There's a new drug target for treating cancer and viral infections. And a new partnership has created a cell-based mRNA design and manufacturing platform. Champions Oncology has launched a radiopharmaceutical services platform. Angelman syndrome results were published. And IGI and AbbVie have announced an exclusive global licensing agreement for a tri-specific antibody. Arcus Biosciences received orphan drug designation for its pancreatic cancer treatment, and you can read all of these and more at deeptechdigest.com. Alright, it's time for the first of today's guests. First, we're talking about neutrophil extracellular traps, otherwise known as NETs, and their potential as a new therapeutic class being developed by Citryll. To tell us more is Citryll CEO Eduardo Bravo. So I guess the first question then is if you could give us some details about the company.
Yeah, so Citryll is a Dutch private biotech company. It was started 10 years ago. We just had a couple of weeks ago our 10-year anniversary, all based on the discovery by one of the founders and still the CTO of the company, Renato Chirivi, who discovered this monoclonal antibody that was capable of modified inflammatory status of animals at that point by targeting NETs, which is something that no other company nor compound was able to do in the past. So that's how the company was started. And I arrived in the company a couple of years ago. I've been many years in big pharma, then in other smaller biotechs, and joined a couple of years ago to help do the Series B, which would close November of last year, and hopefully to help with the development of the company moving forward.
And you mentioned that you're in Madrid, but they're in the Netherlands.
Yeah, the company is based in Os, which is a small town in the I would say southeast part of the Netherlands. That's where this is a little bit of an odd place, and we get asked a lot of times. But that's, for example, where Organon was started, and today in those buildings, Biopark has been created, and there's a big number of biotech companies, some pretty well known like Acerta, and that was acquired by AstraZeneca a few years ago. So it's a very nice research park with very good facilities. So we're happy there.
So you mentioned NETs. I wonder if you could explain what nets or neutrophil extracellular traps actually are?
Yeah, 30 seconds on the background. So neutrophils are, the most common white blood cells that we have, and they are our first defense when the body gets a host attack. And one of the ways the neutrophils have of defending the body is releasing these traps. It's actually they explode and release their content that is inside the neutrophil. And these nets are web-like structures, which are composed of DNA, histones, and antimicrobial proteins. And those nets have been demonstrated to be very pro-inflammatory. They actually produce the inflammation. And, that's what we try to control with the arrival of CYP13.
Right. Well, what's the challenge been on working with nets?
Well, the challenge is that it's a new mode of action. No one has ever targeted nets. So the first thing is convincing actually the third parties that nets were really, nets were discovered just 20 years ago, which, looks like a lot, but in science, this is yesterday. And there's been a lot of debate, and still I believe that there's people that believe that nets are more the consequence of inflammation rather than the drivers of the inflammatory process. So when you're a small biotech in a small European country and you come with something that is completely new, that targets for the first time a new component of the innate immune system, a lot of people were very intrigued, but they feel that there's not enough data to support that targeting nets and reducing nets will have a significant effect on the clinical endpoints. That's our goal.
All right. So there's still a lot of work being done on that?
The number of publications now is exploding, and there's very few inflammatory areas or even related areas where we don't see publications coming with the role of nets as being not only present, but actually causing the inflammatory process of those diseases. So that's a good news. What we're doing ourselves is go deeper. Once we understood the mechanism of action of our antibody, and this is the beauty of our antibody, we can stop the release of new nets, which is great. But most importantly, we can help clear the excess accumulation of nets in the tissue. And we believe that second part actually is probably driving the majority of the effect. What we're doing now, once we understood that and that got published, now we're really digging deeper into why the clearing of these nets actually has this anti-inflammatory effect, and what are the impact, for example, on macrophages that help get rid of those nets once they have immune complexes formed with our antibody. So there's a lot of work that we're doing there. There's additional work that we're doing to elucidate some of the mechanism by which nets are pro-inflammatory. And then hopefully with our clinical trials, we will be able to make the link between impact on the nets and relief of clinical symptoms. So that, of course, we will know once we have our clinical data results.
Because it's something that's so new, how did you approach this?
With a lot of patience. So I think that, there's so much you can do. So it's actually, first thing is making sure that, we devote the time and the effort and the money to the things that will really move the needle, at least our understanding. And then we've been very lucky with the syndicate that participated in the funding of the company. So we have, 2 pharma companies, J&J and Novartis, plus some very well-known VCs that join Forbion and Pureos, that join our existing investors, mainly PGV and Seventure. So there's a lot of knowledge as well on those board members in making sure that, we move on the one end, that we get a deeper understanding, but at the same time that we move as quickly as possible into clinical readouts. At the end, maybe the science will be beautiful, but as the English or the Americans say, the proof is in the pudding. So what we need to see is that indeed in patients, injecting CYP13 by reducing those nets will have a clinical impact. And that's the goal of the 2 phase 2As that we will start.
Maybe you could go into a bit more detail about CYP13 and what it does and how it works.
Yeah, so basically CYP13 is a monoclonal antibody. So, and what it does is go and target those neutrophils that are about to rupture, and just at the very last minute before they rupture and they release their extracellular traps into the outside world, the membrane become permeable. And the fact that we have a bivalent antibody that targets two different histones, that can cross-link at the membrane level and actually allows the neutrophil, which is dead, but not to explode. So at that point, that becomes a dead cell that is then cleared by macrophages. So that is what we've demonstrated also in humans, that we can that way avoid the release of additional nets into the bloodstream and into the tissue. And then the second part, which I said is probably the most important one, is, it's an antibody that will recognize those histones that are present in the nets. So the antibody, and again, we've demonstrated that, does very quickly recognize those histones and attach to these nets. And again, because it has an active Fc tail, the antibody, then those immunocomplex that are formed by the nets and the antibody are cleared by macrophages. So it's this dual mechanism of, we stop new nets being formed, we clear the existing accumulation of nets, then we reduce, we believe, the biggest cause of inflammation happening in the patient. And if you look at the amount of diseases that have a inflammatory component, that gives an idea of what we believe could be the potential of CYP13, which is, pretty phenomenal.
And what kind of conditions are you initially looking to be able to work on with this?
We've chosen, for the time being, two indications. One is rheumatoid arthritis. The other one is hidradenitis suppurativa, which is until not very recently completely unknown, but there's new drugs that are coming on this market. So now more and more people are aware of the existence of this hidradenitis suppurativa. Why we've chosen two? For various reasons. One is to show the breadth of the activity of the compound. Of course, if we could have done more, it would have been even better. But the fact that one is more RA is more an autoimmune disease where, the inflammation happens because of these autoantibodies that have been generated, while the hidradenitis suppurativa is almost a pure neutrophilic disease. So it's a way of showing that the product will have the ability to work in a wide variety of inflammatory diseases. And also, in the case of RA in particular, is a disease where the efficacy thresholds are super well understood and the clinical endpoints are undisputed. So showing efficacy in a disease where, everybody will be able to put our data in comparison with other compounds that have been tested on endpoints that have been used for many, many years and are very easy to follow, we believe that will give a very clear answer to the world on the level of activity of CYP13 in those patients.
Where are you at on the pathway with clinical trials?
So we've done all the animal work. We've done the safety part in volunteers, both healthy volunteers, but also some RA patients that volunteer to be dosed with CYP13. And we've demonstrated that the product is well tolerated and there are no side effects associated to one or two doses of CYP13. Now, of course, the real challenge starts as we're going to be moving into large controlled trials in which patients will receive either placebo or different doses of CYP13. And we're going to be looking at, in the case of RA, it's a 6 weeks endpoint. In the case of HS, it's a 12-week endpoint. And we're going to be looking at the most traditional endpoints for those disease areas and looking at the difference between, giving a certain dose of CYP13 or placebo. So a lot will be learned. We'll start recruiting on those trials probably just before the summer. We expect to have the preliminary results on both indications by the end of next year.
And what's the mode of delivery for the product?
So today is a subcutaneous injection that needs to be injected every other week.
All right, very good. You don't have any results until next year, but what are you looking for as being success for both you and ultimately for patients?
Yeah, so we have data on very few patients, and that data will be presented in ACR this year in October. So some of the healthy volunteers that participated in the clinical trial where we did repeat dosing, actually we have some interesting activity data in those very few patients. So that will be presented at ACR. So we're not disclosing those results. But now when we move into the large clinical trials, what we need to demonstrate, of course, is superiority versus placebo. That's the first thing. But I think that in particular with RA, where the endpoints are very well known, I think that people will be able to look at efficacy compared with existing therapies. So we expect to see at least efficacy as good as any other product that has been made available on these indications.
And is that something that people would need to take constantly, or would they be able to stop taking it after a certain point?
We would love to know the answer, Jim, of those questions. That will come with the data. In principle, because probably that's what has happened with the different mechanisms that people have developed up until now, we are assuming that this will be a chronic administration. Now, it's true that in some of these diseases, you get outburst of the disease. So if there's correlation between those outbursts and NETs, we could either have, a very low maintenance dose, or we can have a product that will be administered on the outburst. But for the time being, we're treating all the patients for a small number of weeks, and we will continue developing it for the time being with the knowledge we have as a chronic therapy.
Right. You mentioned being a pioneer in this field. Are there other companies that are looking at NETs?
Yeah, there's two companies. There's a company in Europe looking at cleaning the blood with a sort of a dialysis system out of the nets, but that's not as convenient as you can imagine. You need to go into the hospital. You need to, get all your blood being pumped out and in back again after having been filtered. So, but for very severe cases, it may be an option. What we believe is a great proof of principle that eliminating nets from the blood and from the body produces an effect on severe patients. So that's how we see it. And the other company has another way of looking into the damage that is produced by the nets that does not target nets specifically. So we're so far that we know the only company that has a monoclonal antibody that is capable of targeting in such a precise way nets.
Do you think it's a field that we'll start to see more activity in?
Well, I think that people are going to be looking at our clinical data, which is what happens when you're the first one. Everyone is like, great, show me the data. If we prove, and as you can imagine, we're not objective here. We're convinced that we have something that should work. This is a completely new way of treating inflammation and way up what I would call on the inflammatory cascade. So we think that this, honestly, if it works, and there's a big if there, but if it was to work properly and indeed the nets are demonstrated to be the true initiators and the main responsible things of the inflammation, I think that this has the potential to become, an incredibly big compound that then I'm sure that people will follow, like with the PD-1s, like with the anti-TNFs, but we're looking at that sort of compound. Because
of that newness, has that reflected in fundraising?
Yes. Yes. We were very lucky to actually close our financing round in November last year, because if anything, the market has gone worse. But even then, it was not easy. And the main difficulty was the novelty. One would expect that having a first-in-class will make investors more interested. And I think that to a certain point they were. The amount of investors that, signed a CDA, went into the data room. But I think that at that time, because, we're talking with those investors now more than a year ago, we didn't have any patient data. Now, at least we have a few that have received the compound. What I said at the beginning, what we call the chicken and egg issue. Are the nets the consequence, or are they the drivers of the inflammatory process? We're convinced. We think that there's a lot of literature that shows that, they are the drivers. But then convincing people to risk the money that by targeting those nets, it will have an effect on the clinical efficacy was challenging. And, it was a very hard process. Very happy with the syndicate. I think that we have an amazing syndicate. And so at the end, it was good that it was tough. But I tell you, it was long. It was long and painful.
I can well imagine. Yeah. It's great that you're doing something very new, and there's obviously a lot of work involved with that. But are there other things that you're working on alongside of this?
No, I think that when you have something like this, it's very difficult, in particular in Europe, which is, has I believe, a more cautious approach into developing companies. I think that we have our hands full. I think that we could spend another financial round in doing additional things with SID13 rather than jumping into something else. I mean, I think that this new mechanism could be so big that it deserves full attention, and we need to see and demonstrate. That's what they've given us the money to do. Is does it work or not? We just need to do the trials, recruit the patients, do it properly, and get the readout. If we are right and it works, I think that is a huge finding. I think that is very exciting times. That is, an amazing year and a half on something that is very new. The market need, I mean, there's many products that have arrived in this, with these biologics, the RA patients for HS, for psoriatic patients. I mean, there's many things that today work, but it's still I would say at least 30 to 40% of the patients are not controlled properly. So I think that there's a huge need, and that's our goal, is to bring to the patient something that may alleviate them from something that today cannot be alleviated with what exists out there. And rather than coming with another one, we believe that working on something that is so unique and new is what drives ourselves to try to get there as soon as possible.
Well, that's an exciting approach, and always interesting when a company is blazing a new trail. Even though I live in Scotland, we're in the southwest, and our closest capital city would be Belfast. So that's where we're headed next, to learn about antimicrobial resistance and using AI to develop new drugs. The company concerned is AMPLY Discovery, and to fill us in with all the details is AMPLY Discovery COO Dermot Tierney. Thanks for joining us, Dermot. I wonder if first you could give us a bit of background on the company.
So thanks for inviting me along. Amply, to explain what we do hopefully very quickly. Amply is a tech bio startup. We're based out of Belfast in Northern Ireland, and we spun out from a university in Belfast called Queen's University in 2022. And we have built an AI-driven drug discovery platform, which is a little bit like ChatGPT for molecule discovery. So we use machine learning and synthetic biology to discover new therapeutic molecules. And the aim of Amply is to tackle some of the toughest health challenges. So, for example, drug-resistant infections through to potentially even complex cancers. And what makes us unique is how we harness evolutionary biology alongside AI. And so we comb through these massive genetic and genomic data sets. So, for example, patient omic data, metagenomic data, transcriptomic data, and we can biomine this data. We can find promising new molecules or interesting potential drug targets that nature has evolved usually, but that humans have not yet studied very well or characterized very well. And so our starting point in the university through our research was focused primarily on something called antimicrobial peptides, AMPs. These are small proteins that can typically kill bacteria in very novel ways. And so since our algorithmic approach can identify these potential drug candidates digitally, we could then add synthetic biology to that whole mix, and we could synthetically produce or bioprint those molecules that we predict. We test them in biologic assays. And ultimately, we believe that this joined up approach can help us discover new drugs faster. And so at the moment, we focus on two flagship R&D programs that we're working as really our proof of concept for the platform. One is around RNA interference therapy, where we mainly work in cancer. And the second is our inhaled antimicrobial work for antimicrobial resistance, where we're working against drug-resistant lung infections. So this would include, for example, drug-resistant TB, or CF-associated lung infections. And we're pretty early in the journey, I would say. We're startup stage, we're a seed stage company. We've raised around 4 million US dollars to date through a mix of sources. And this has helped us make very rapid progress in assembling a pipeline of novel drug candidates and then demonstrating that this AI-guided, evolution-based approach can actually work and potentially deliver unique candidates that maybe other people might miss.
With regards to AMR, could you maybe go into a bit of detail about what that issue is?
Yeah, absolutely. So antimicrobial resistance is what it stands for. And it's often called the silent pandemic. And so in very simple terms, AMR is what happens when microbes, like for example, a bacteria, but also potentially a virus or fungi or even a parasite, these creatures effectively evolve over time to resist the drugs that we would normally use in them. And so over the years, since antibiotics have been developed, around 100 years ago, over that time, the misuse of antibiotics has helped bacteria develop defenses, and even just the general use of antibiotics. It's not simply attached to misuse, just even the general use of antibiotics allows over time bacteria to develop defenses so that these antibiotics no longer work. And what this means in practical reality is that common infections that used to be really easy to treat can suddenly become deadly again. And it's a growing problem. It's now, it is considered one of the top global public health threats. To put it in a little bit of context, over the last few years, there are about 1.2 to 1.5 million deaths worldwide directly associated with bacterial drug-resistant infections. But also there are up to around somewhere like 5 million deaths that are associated with some type of AMR. So when bacteria like E. coli or staph, or even more complex bacteria like TB become resistant to these drugs that we use today, then quite simply doctors run out of options to treat, and patients can potentially die. And I think the thing that AMR is such an overused term, I think people have become sort of slightly impervious to it, and it's so slow moving. It's unlike, for example, the COVID pandemic that was there. It was immediate. It was government reactions. AMR is very slow moving. It moves with evolution. But ultimately, I think what we need to understand is it doesn't just affect a few rare diseases. It threatens the entirety of modern medicine and potentially the very basis of society that we're living in. So if antibiotics don't work, even routine surgeries or chemotherapies become riskier, and because the threat of an untreatable infection looms. So if you think about procedures today that we take for granted, so like a C-section or potentially an organ transplant, that are becoming increasingly straightforward, but these rely on effective antibiotics or antimicrobials to prevent and treat infections. And so if a drug-resistant superbug spreads, then it undermines our ability to perform these, safely, even these basic types of procedures. And well, a big driver, as I mentioned, of AMR is misuse of conventional antibiotics. This, for example, is sometime everyone uses an antibiotic incorrectly. Evolution also just on its own plays a big part. So time, but also the misuse gives bacteria a chance to adapt and become drug resistant over time. And so when you combine that with things like heavy antibiotic use in livestock, where a lot of the time we're using the same types of antibiotic in livestock that we need for humans, maybe poor infection control in certain settings, what's happening here is you're creating a perfect storm for resistance to spread globally, and it is spreading globally, and it's affecting some of the developing world first, but it's increasingly affecting the more developed westernized healthcare economies. I don't want to be too hyperbolic about it, but we're figuratively standing in the doorway of a time machine, but the worst kind of time machine, one that is going to take us back 100 years to a period where antibiotics just simply don't work anymore. Infections get harder to treat, and that will set us back and set medicine back to this, what we consider to be the pre-antibiotic era. And if we believe some of the predictions, and there is lots of reason to believe these predictions, for example, a very strongly cited UK government report predicted that by 2050, 10 million people would die or be at risk of death due to drug-resistant infections. So that's obviously a significant driver of the unmet need that exists with AMR.
For many years, it's been known that this is an issue, and there's a lot of research. What are companies and researchers doing to find a solution, I guess?
There's a ton of innovation actually within the AMR space, but the unfortunate thing is there's not a commensurate amount of funding. And so researchers and biotech companies like ourselves, we know that we need new strategies to outsmart these drug-resistant bugs that I've mentioned, because these old antibiotics are failing. And so you have companies that are working on new approaches and classical discovery techniques for new antibiotics, conventional antibiotics, small molecules from various different sources. This is exceedingly slow and exceedingly expensive. There are companies like ourselves, including ourselves, working on antimicrobial peptides as a potential source. As I mentioned, these can be natural or synthetic small molecules, and they destroy bacteria through certain mechanisms. We're working on them because we believe they can be highly effective and a key part to treating drug-resistant infections, because bacteria have no pre-existing defenses against them. They can have comparable effect and potentially be quite broad-acting in the same way that conventional antibiotics can. You also have things like phage therapy. So this is where companies and researchers are looking to find bacteriophages, so small viruses that can infect the bacteria. They're very good at targeting and killing certain bacteria, but they're also very specific. And that means you need the exact right phage for each infection. So it's very difficult in that context to develop broad medicines. And other areas, like, for example, antibiotic adjuvants. So these are helper molecules that will block the resistance mechanism and render these conventional existing antibiotics more useful for maybe a longer period of time. Other people are working on vaccine technologies, and other people are working on rapid diagnostics that can identify pathogens quickly so we can understand the specific type of bacterial strain that we're actually working on, and be very specific about how we kill it and what we use against it. And then what is used right across the space is antibiotic stewardship, where people are trying to limit the use of existing drugs or use them more widely so that it slows down the pace of antimicrobial resistance being generated. But what I would say, and what I really want to underline, is that it's a really tough fight, because while all of this innovation is going on, the economic logic of the infectious disease medicines is broken. So when a new type of antimicrobial is created, it does not attract the same sales or the same prices as, for example, a new cancer therapy. In fact, it doesn't attract the same sales precisely sometimes because of the stewardship issue. It's valuable as long as it's not used, but clearly this does not fit well with the economic logic of any commercial company that is there to drive sales. So you have this terribly unweighted market system, and so we're now seeing innovation at the government level trying to change that a little bit with the advent of something called subscription pricing, where governments will kind of subscribe. It's a little bit like a SaaS subscription model, but for medicine. So it's quite unique and interesting, but it's, as you might imagine, when governments are involved in as a key part of moving it along, it's moving along very, very slowly. So that's one of the biggest challenges affecting the market at the moment.
Can you tell me a bit about the approach that you're taking to solve this?
Yeah, so our approach is a little bit unconventional. I like to say that we merge evolutionary biology with artificial intelligence or machine learning. And so we work with antimicrobial peptides, and we don't start with a single target or a single chemical and then do years of trial and error. We have built a platform which is a digital drug discovery studio that scars huge databases of genetic sequences and natural molecules, for example, the genome of a particular plant or maybe a particular animal, that have been developed through years of evolution itself, to find promising candidates. We call this biomining, and we use AI to search through these metagenomic or sometimes genomic data sets. And we predict new drug-like molecules. We're ultimately looking for these little hidden gems, these molecular blueprints that could eventually become medicines. And once the AI has done its work and made these predictions, we can then move that into the lab, where we use advances that have happened over the last decade in synthetic biology to synthetically produce or bioprint these molecules. And then we test them in vitro. So we test it in test tubes, petri dishes, in a lab-based environment. And this lets us validate if these AI discovered compounds are actually having the desired effect, for example, killing a drug-resistant bacteria or silence a particular gene, and then we iterate the process. So the data from those experiments feeds back into the system, and that is used as the fundamental basis to improve the models that we've built. And then we can from there zone in on the most effective candidates. So in short, we've created this design, test, refine loop, and it makes the discovery process for us that much more faster at the discovery end. And the beauty, I suppose, of what we do with the platform is it's not limited to one disease area. We have essentially created a search engine, if you like, for biologically active molecules. And we work on antimicrobial peptides for infections, but the same approach, the same basic approach is working very, very well in long encoding RNA target discovery for cancer applications. And in fact, the technology platform that we've built can hunt for other functional units of biology, and we can tune it to look for inhibitors of inflammation, anti-environments, or even novel cancer targets. But of course, the issue for us as a small startup with a relatively small amount of money is we need focus, and we need to focus our resource in a certain area rather than trying to boil the ocean. And that's why we've zoned in on the two core pillar programs that I mentioned, where we have one that's working on antimicrobial resistance and drug-resistant lung infections, and our other then that is working on the discovery of gene silencing RNA therapeutics. And so we think the platform is very versatile and can be pointed at potentially any disease where there's ultimately a biological target. And we hope that the programs that we're currently running will demonstrate the power ultimately of the platform.
And how do you get basically from scratch using your platform on a pathway to a drug?
Drug discovery itself is a very, very long process, and we really operate at one piece of that process, which is the discovery side. So going from scratch to develop drug is long and pretty arduous. What we do is first we define the problem. Say we want to kill a particular type of superbug or find a molecule to silence a particular cancer-related oncogene that we've identified. And so we start with that goal, and then the AI platform will generate a slew of potential molecular solutions in silico, so digitally. For example, with our MDR bacterial infections that we work on, the system might propose thousands of antimicrobial peptide sequences that could attack a particular pathogen in different ways. And we will use metagenomic data as a starting point for that. So identifying these sequences that have been evolved through years of evolution, maybe in a plant or a particular animal to fight that type of pathogen. And then next we take our top ranking candidate molecules based on those predictive models. We make them. Modern techniques allow us to essentially to print out peptides or genes that correspond to these types of designs. And then we can put these candidates through preliminary testing. So in the case of an antibiotic, we quickly check, do those molecules actually inhibit or kill the bacteria that we want to kill in an in vitro assay. And then we also make sure that these aren't overtly toxic to human cells at this stage. We test for lots of other characterization data too on the molecule. We might want to know a little bit about its biophysical properties. So for example, its solubility or its charge, or run tests around its stability. And so the system predicts all of these factors, and we feed all of this data back in based on its predictions. And we loop this back in to create an iterative cycle. Once that molecule looks promising in vitro, and we've gone through typically rounds and rounds of optimization, we might then start introducing more rational design approaches where we will tweak structure. We'll do single amino acid substitution to improve things like stability, for example. And we continue this loop until we have a top candidate that performs well in those wet lab tests. And then we can move through to our preclinical studies, which often means that we will move it through to an animal model and see how it fares in a living organism and to gather initial safety data. Again, using our TB program as an example, we would test it in mice to confirm if it clears the TB lung infection and also doesn't cause harm. And so then success, obviously, in an animal study will give us confidence that the data can be moved through then the next traditional steps of moving to IND enabling studies and ultimately then into eventually clinical studies. So I suppose that's a very long-winded way of simply saying that we start with an AI design, we swiftly make and test these designs in the lab, we refine them and perfect them through iterative cycles, and then we advance the best candidate into preclinical, and we hope eventually into the clinical development. So it's a very, very careful and still a stepwise process, but using AI at our phase, at the discovery phase, And the rapid testing piece means that we can go much more quickly from a digital idea to actually a tangible drug candidate.
And what's your kind of business model? Is it to do the clinical trials yourself, or is it to approach companies with a solution?
Yeah, so at this stage, we're moving programs through ourselves. We're picking the problems ourselves and then developing the solution. So rather than waiting around or pushing towards a big pharma company where we're at the early stage of validation, we consider the AMPLY platform to be a drug discovery studio. And that means that we internally incubate these new therapeutic programs. So for example, we identified drug-resistant lung infections as a critical area where there's an unmet need where our platform could add value. Similarly, on the RNAi side, we identified, specific, highly genetically driven types of cancer like acute myeloid leukemia or triple negative breast cancer as areas where our platform could find novel RNAi-based drugs for that. And so in each case, we weren't contracted by an outside company or pharma company. We proactively selected that challenge or that problem, and then we went about solving that. That said, our approach is ultimately very collaborative by nature, and by nature of a startup, you want to eventually work towards larger pharma partners. So we envisage eventually working with pharma partners in one of two ways. So approach number one is where we take those existing programs and the assets that arise out of those programs, and we partner those after we have preclinically validated them. So we have taken on a portion of the preclinical validation risk. We have validated that this could be an effective medicine, and then we partner that with a partner into the clinic. This is obviously very helpful for us so that we as a sort of a preclinically focused startup don't also have to then similarly try and build up a complete clinical infrastructure and clinical team. And then secondly, and I think eventually as our platform progresses, as our own programs move through the development stage, and as we generate more validating data, we do envisage then working with pharma partners at an earlier stage where pharma partner or a research institute come to us and say, we need a drug that does X. Can your platform help? And then we deploy our technology to find that solution, acting as an early stage discovery partner. So we're open to both avenues, and there's no real one size fits all here. In practice, we're focused at the moment on building our own pipeline of good candidates because we need to ultimately prove out the tech and also create value. But then success on those will naturally breed, I think, interest and potential further down, opportunities for downstream partnering.
What successes have you had so far in the pathway that you're taking?
So consider we're pretty young. We have hit some nice, exciting, albeit early stage milestones. So one big success that we've proven out actually the core concept that actually the AI plus biology platform can actually discover real candidates. So over the last couple of years, we've used it to generate multiple promising molecules, particularly in that infectious disease space. So we've identified novel peptide candidates that show potency against drug-resistant bacteria, TB, for example, and also CF, cystic fibrosis associated lung infections. And this kind of validation, this preclinical validation that we're getting is a crucial step for a startup like us. It sort of demonstrates that we are not just a completely in silico tech bio team, a team full of hoodies, guys in hoodies, that we actually can translate this through to a wet lab environment. And then other successes, tangible successes also in the support and the funding that we've attracted. So since we've spun out, as I mentioned earlier, we've raised about 4 million from various sources. That has helped us with our funding and development to date. And that has also allowed us to build out a strong team and strong partnerships. So we emerged from a purely academic setting in Queen's University in Belfast. And then since then, we've brought together a really strong team of experts with machine learning, molecular biology, drug discovery expertise, and we've also then collaborated with institutions and biotech partners. So, for example, we've worked with St George's University Hospital in London, who have a very strong TB team and TB program. And we've participated in accelerators like, for example, Techstars, which is an international program and has helped us sort of expand our network and achieve sort of international exposure. And lastly, I suppose, but most importantly, really, is the pipeline itself. While it's early and it is at the preclinical stage of development, we're currently seeing success. We have 2 lead programs underway with the RNA therapy in cancers and the inhaled antimicrobial work in drug-resistant lung infections. And both of these now are progressing through the preclinical development stages. And our ultimate goal, probably over the next 12 to 18 months or so, is demonstrate our platform's versatility by tackling these very different challenges and actually these very different modalities. And we're on track in terms of that. So if we can advance these through to the next stages beyond animal studies and eventually into the clinic, then that'll be a huge success for us. So I think even at this early stage, we've built a functioning discovery engine, and we're seeing the early shoots of success in that process.
Not just thinking about AMR, but in general, a lot of companies are using AI and applying it to drug discovery. When do you think that is going to come up with some potentially blockbuster results, seeing as everybody seems to be thinking AI is the future of drug discovery?
Yeah, so it's true. There's a lot of talk and, dare I say, hype around AI and drug discovery. And I suppose I think it's reasonable to say we're seeing the early fruits of this approach. But drug development is a long game, and what I've talked about very much is the preclinical side. But our technology does not necessarily, well, it makes the discovery side more predictive and quicker. You still have a number of steps to go through once you need to go through the regulatory steps and get clinical trial approvals. So it is a long game. It's going to take a while to materialize. To kind of give you a sense of context, the first AI designed drug candidates entered human trials sometime around 2020. And right now there are multiple at this stage AI discovered or designed molecules that are in either phase 1 or phase 2. In silico medicine have one that's in phase 2, for example. It's probably going to be the latter half of this decade, so 27, 28, before you see those drugs ultimately go through that whole development process. And then to find a blockbuster, that's a different challenge again. So a blockbuster is something that actually is not just approved, but it becomes widely used and has a kind of a massive impact. And so that, you're talking a few years later, so maybe 2030. But one thing I would say in answer to that question as well is I would also ask a question. And the question is fairly simple: is what is an AI drug? Is it a molecule design? Is an AI drug a drug that has somehow used AI in the clinical trial process? So there's so many parts of the drug discovery process that are now using AI that I think by the time that we end up in 2030 with our first end-to-end AI drug, we won't even be thinking in that way. We're not going to be thinking in terms of AI or not AI because it will be such a ubiquitous part of the process. It's just another tool, just like the internet had huge hype around it at the time, and now it's pretty ubiquitous. We take it for granted. Electricity, when it was invented, went the same way. I think this is where we're getting to. Drug discovery is a huge challenge for AI, and it's probably one of the most difficult challenges. It's a much more difficult challenge than, for example, large language models. And the reason is pretty simple. I think we all understand that we know a lot about our own language of all forms and sizes by now, but we're still at the very tip of what we actually understand about biology. And so as we understand more and more about biology, as we can drill deeper into what makes us us, then AI is going to be even more valuable in terms of mining and understanding and quickly getting to the bottom of all of these masses of new data that will arise. So as far as I'm concerned, where AI has value is speeding up the early stages, and that's where we've focused, finding new candidates, validating new drug targets much faster than we've been able to do it before. And it has allowed a very small company out of Northern Ireland, which is a very low capital market but high knowledge intensive market, to make progress with a small pot of startup funding. There was a time not that long ago where you took your 4 million for drug discovery, you wouldn't have got much change out of your small molecule screen. And now we're building an entire company with multiple different drug discovery programs and advancing it through preclinical. So AI has allowed us to exist and also compete with companies with much larger resources.
Well, unfortunately, antimicrobial resistance isn't really a big ticket item, but as Dermot mentioned in that interview, it's a massive issue and it's got huge implications for other areas. We often look toward the future and things like AI, but we don't really want to head back in time to when there were no antibiotics. Anyway, that's it for another podcast. If you have questions or comments, suggestions for guests, or want to sponsor one or more podcasts, please do get in touch. And you can do so at jim@deeptechdigest.com. So, until next week, wherever in the world you are, I hope you have a great week. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.